What's up, Dragonfish? Welcome back to Rock Bio. Today, we're going to look at meiosis, one of the types of cell division.
All right, first we covered mitosis. Mitosis was an example of asexual reproduction, resulted in two cells that were both diploid. Sexual reproduction pertains and relies on meiosis. Meiosis creates our gametes or sex cells that are haploid. All right, we have, as I mentioned, body cells that are also called somatic cells made by mitosis. Those cells are all diploid. In humans, we have 46 chromosomes, basically 23 pairs. The sex cells, also known as gametes, include our eggs and sperm. Those are made by meiosis. They are haploid. At the end of meiosis, we end up with 4 cells that are haploid and that are genetically all different from each other. There are 23 chromosomes in each gamete, and therefore they are called haploid. Here you can see the sperm are a good deal smaller than the egg. Of course, it only takes one sperm to fertilize an egg. Then when we look at fertilization, we end up with a zygote. Remember that each gamete, each egg and sperm are haploid. So when the fertilization process occurs, we have a diploid cell. So at that point, we have 23 chromosomes from the egg and 23 from the sperm, giving us 46 chromosomes. That 46 number is 2N, also known as diploid. Meiosis is basically considered reduction division because it starts with a diploid cell, it replicates, and then there are 2 actual divisions that occur that allow for 4 genetically different haploid cells. Basically, we have 2 steps. There's meiosis 1 and meiosis 2, PMAT1 and PMAT2, if you remember, or PMAT1 from mitosis, and we had PMAT2 for meiosis.
Okay, so we're going to start by talking about meiosis 1 first. So you have a prophase 1, metaphase 1, anaphase 1, and telophase 1, or that PMAT1. Homologous chromosomes are these non-sister chromatids that are going to be divided, and you're going to have 2 new diploid daughter cells. So very similar to mitosis here as far as what we're actually producing. Prophase 1 is one of the most important steps because the chromosomes are going to coil up like in mitosis. And what's going to happen though is they're actually going to form this tetrad, or they're going to form together. We have 2 sets of chromosomes lining up next to each other. And then this process called crossing over can take place. Crossing over, also known as genetic recombination. So sometimes while they're next to each other in that tetrad, parts of the chromosomes will overlap and they will actually exchange genetic information with one another, which leads to 2 chromosomes becoming more different, or more variations in each chromosome as they get passed on. So you can see this in the picture here. We start out with a single pair and they form the tetrad. They overlap and that crossing over occurs. And so you get a little section from the chromosome here and a little section from that chromosome on this side. And you wind up with 4 at the end that are all different from one another. That adds to variation, which is a good thing. Metaphase 1, all the metaphases are pretty much the same. The tetrads are moved to the equator or the middle of the cell. They're going to line up independently of one another, so they're not predetermined where they're going to go, but they line up independently in mitosis, and they're going to line up in pairs in meiosis. So that's a little bit different. Anaphase 1, exactly the same as normal anaphase. They're going to get pulled apart, except for the chromatids are not going to split. They're going to travel as pairs to each side, so they're not actually going to split in half. You're moving the whole pair to one side of the cell or the other, and then they're going to actually form 2 new cells in telophase 1. Cytoplasm is going to divide, and so each cell has one of those homologous pairs, and they're still attached by the centromeres. So you have two diploid daughter cells. That moves us into meiosis 2, where those two daughter cells are now going to each go through prophase 2, metaphase 2, anaphase 2, and telophase 2. The sister chromatids are now going to get split, so they get separated, and they will form 4 new genetically different haploid cells. So meiosis 2 is very similar to mitosis as well, and that the chromatids are going to separate now. So prophase 2, a new spindle is going to form. Metaphase, they line up in the middle again. Anaphase, they get pulled to opposite sides, just like before. Telophase, the spindle fibers dissolve, you form new nuclear membranes, and the cytoplasm divides, leaving you with those 4 cells. So here's a really simplified diagram. Basically, you're starting with those pairs, you divide the pairs into 2 new cells, and then we divide those pairs in half into 4 new cells that each have half of the genetic information. Here's a different picture with a little bit more illustrated visual. So we're going from prophase 1 through metaphase, anaphase, telophase 1, and then we have 2 new cells. Those 2 cells each go through the second phases until you get 4 new haploid cells. Remember, these 4 cells are all genetically different. They're not the same. One important difference to note between egg cells and sperm cells is whenever you're producing sperm cells, all 4 sperm cells are going to mature into sperm cells that will be used. Whenever you're forming egg cells, the best egg cell is chosen to become an actual egg cell, and the rest of the cells are just going to become polar bodies, and they'll actually be used for some other things later on. But you're only going to be using the best cell, and that's about creating the best possible offspring. This is probably the most important information here for you to look at, comparing the two. Just a refresher on mitosis, you're making 2 cells in mitosis. Those cells are body cells or somatic cells. They are genetically the same, and they are for growth and repair. That's in contrast to in meiosis, you're making 4 cells. All of those cells are sex cells, also known as gametes. They're all 4 genetically different, and they're used to form the sex cells for genetic variation so that organisms are not all alike. We're not trying to create clones, we're trying to create different organisms. Please be sure that you write down any questions that you may have for your teacher so you can ask them in class.